This is the CRISPR Help Session Part 2. So, as the name suggests, this is the final installment of our CRISPR Help Sessions. If you weren't able to join for Part 1, I highly recommend you watch the videos from our website. We had three breakout rooms operating at once on three different topics. We had guide RNA design, vector cloning, and plant transformation. And today we will be discussing analysis of edited lines. So please do keep your questions on the topic of edited lines. You may ask questions in the Q&A box, so you can find that at the bottom of your screen. There should be a little box with a question mark. That is where you want to ask your questions. I will be moderating, so I will be reading through the questions and reading them out loud to our speakers who are going to have a chance to answer. And if there's a question you are particularly interested in, you can like that question. So if you want to see something, if you want me to read something out for the speakers, feel free, go ahead and hit that like button, bring my attention to it. And additionally, if you would like to share your own experience and expertise, you can comment on the question. So with that, I'd like to introduce our speakers. We have CJ Chai and Josh Clem. CJ is a Winfred N. Haynes Professor at the University of Georgia. She is passionate about trees and is especially interested in their longevity. She uses a variety of approaches in her research, including CRISPR genome editing. And she is the creator of AGE-seq, the first open source software available for analysis of data from high throughput sequencing. Josh is a PhD candidate in Dr. Yiping Chi's lab at the University of Maryland, College Park. Since studying molecular biology as an undergraduate at Frostburg State University, Josh has been fascinated with plant biotechnology and its utility in improving crop productivity into the future. Josh decided to join Yiping Chi's lab at UMD to focus on plant genome editing technology development, where he leads several projects on novel CRISPR-Cas systems and reproductive trait engineering. He also mentors several undergraduate research assistants in the Chi lab. Great. So again, just thank you so much to our speakers for being here and sharing their expertise and their time with us. Again, you can answer or enter your questions in the Q&A box. And I'm going to start us off with some questions that you all submitted during the registration process. So we are going to start with, what are the most common challenges you face when analyzing plant mutations in CRISPR experiments, and how do you overcome them? CJ, I'll start with you.
Sure. I guess I should just preface that we work with Poplar through organogenesis. So our editing efficiency usually is pretty high and we get biallelic edit as the norm. So the most common problem is when we don't get biallelic editing. Typically when we target multiple genes or gene duplicates. I know some of the audience will be interested in polyploids and such, even though I don't work with them. When we deal with gene families, duplicated genes, the situation is very similar, especially for tendinly duplicated genes. So the solution is to use more than just one method. Sometimes we have to kind of design assays in multiple different ways. And then the other thing is mismatch of the reference sequence. We think we design a guide RNA or having the assay against a reference sequence. If that is not accurate, often cause a lot of analytical issues downstream.
That was something we talked a lot about in our part one. Was a very common mistake is people use their reference genome and don't sequence their actual plant material. So again, that's very important. Sequence your plant material. Don't rely exclusively on the reference genome. Josh, how about you? What are the most common challenges you face when analyzing plant mutations?
Yeah, so I mean, just going back to kind of starting my PhD here, I'm not a bioinformaticist, right? So I'm kind of more of a wet bench scientist, and I've trained myself in bioinformatics to the extent that I can analyze my, genome editing results, but I'm certainly not a bioinformaticist. So when I first started, and I would get these like enormous sequencing files because we typically do NGS, Amplicon NGS in particular. And I was like, what the heck do I do with this? How do I actually parse this out and wrangle the data and then visualize the data? And there are many different online resources to kind of allow you to understand what you're doing, whether you choose to use like a sort of a web-based service, or if you work in the command line. I mostly work in the command line now because you're typically able to adjust more parameters if you work in the command line. Um, but nowadays, and this is different from when I started, you have tools like artificial intelligence, in particular ChatGPT, that can really help you to learn to code to some extent, and how to wrangle and manipulate and visualize your genome editing data. And so I would highly encourage people, when they're having trouble in dealing with their just raw NGS reads, and what's the next step. Obviously, don't rely too heavily on AI. You want to be learning in the process, but AI can seriously help you learn the process of dealing with raw sequencing data, and in a way that can be more beneficial than having, like, maybe a postdoc in the lab kind of teach you step by step. And then the other thing that CJ pointed out, a different problem that we faced, is also not achieving biallelic mutations. So oftentimes, you'll look at your sequencing reads, and you'll see a number of mutations. You might see a high percentage of wild type, or maybe you'll see one copy that's seemingly edited, or you'll see kind of a mosaic of genotypes, right? Broken down into different percentages based on those reads. And so we typically, the way we deal with this is we consider, if you're looking at sequencing reads, we look at it, and if more than 75% of those reads contain mutations, we generally consider them to be biallelic. If if the mutation frequency ranges from, say, 25 to 75%, we would consider that to be monoallelic. And if it's below, say, 25%, like in the 10 to 25% range, we kind of consider that to be mosaic or chimeric. And then anything below 5% is oftentimes just a result of sequencing error and or contamination. Um, so that's kind of a couple of things that are challenging to deal with, both technically and conceptually. But yeah, so we've got some workarounds there. So I'm happy to talk about that or answer other questions as well.
Great. Thank you both for that. Our first audience question is from Avinash. How do you detect the gene edited plants that are homozygous if the edited allele is recessive and will not be expressed in the plant? So, I'm reading between the lines here. I'm assuming this is about if you're not able to phenotypically determine, how would you detect your edited plants?
I could jump in. Editing occur at the DNA level, so expression really is not an issue at this stage. But they said, I have worked with colleagues who nominate genes based on, for example, GWAS study of a different species closely related to the transformation genotypes. You do need to watch out that the genes present in, for example, Populus trichocarpa, some genes are actually absent in the aspen, Populus tremula, for example. So those will be the first thing that you need to make sure your target genes really are there. Once the target gene is there, determining mutation is entirely at the DNA level. So in my lab, we actually do those analyses as soon as we get regenerated plant. We label them, we do amplicon deep sequencing through Illumina, and then identify the events. Typically, we select 3 to 5, we propagate those, and then we do a second round of confirmation from the propagated individual. So we basically sampling the same event from 2 different plants to confirm. And then the rest, like Josh was talking about, you have biallelic, monoallelic, based on your research question, you select what events they want to analyze. So at this stage, expression is not a consideration. An edited plant depends on your guide RNA design. They may trigger nonsense-mediated mRNA decay, so the expression could be reduced or not, depends on the target genes.
Yeah, so I'm pretty much on the same page as CJ. It happens at the DNA level, so you have to go in and actually look at your genotype results, and that means some sort of sequencing. There are other, sort of agarose gel-based methods using restriction enzymes like RIFLIP or restriction fragment length polymorphism. However, that can be challenging to assess editing efficiency using methods like that. Moreover, I think CJ and I both rely on amplicon NGS, next generation sequencing, right, as opposed to, say, Sanger sequencing. And the reason we rely on that is because you get much higher depth, which provides a lot more information, sequence information, than you would get from, say, Sanger sequencing, which has more of a detection limit of around 15%. So if you're working with challenging species, like species that are challenging to edit, or you're working with lower editing efficiencies, such as base editing, prime editing, or just, unknown guide RNA editing efficiencies, then it's typically better to resort to something like NGS to detect those potentially very low editing frequency results. Um, so you have to rely on some sort of sequencing, I would say, and the same for us. We often aren't able to detect a phenotype until much later in the developmental stage of the plants. Uh, in contradiction to a gene like PDS, where you would see an albino phenotype if you have a biallelic mutant early on, if you're editing genes that are related to, say, grain size and grain weight, which we commonly use as target genes, you can't even see a phenotype until they start setting seeds, right? So if you want to know if you have an edited plant, it's often better not to just rely on the phenotype, rather the genotype, which requires sequencing data.
Thank you both for that. Speaking of sequencing and detecting the edited plants. So this isn't necessarily related to the edits, but I wanted to talk about how to check whether the lines are transgene free or not. So Josh, maybe we could start with you. If you can give us a little bit of insight into how you check whether these lines are transgene free.
Sure. So basically, the most straightforward way, of course, you can have plants, say in the T0 generation, that are surviving on selection media, and assuming that they're not, escape plants, and we don't really have to deal with escape very much in say, Kataki rice. Um, then you'd have to maybe look into like PCR amplification of a portion of your tDNA. Typically, the way I do it is I kind of overlap, say, Cas9, or I along with the promoter, say, driving my guide RNAs. I basically just try to amplify a section that isn't going to have any sort of unintended or amplification from either the plant genome or some contamination. So I could amplify a section of the guide RNA. As long as you're amplifying some portion of your tDNA, I would say, and you're sure that there's not going to be any off-target amplification, that's a sufficient way to detect transgenes. And it gets more complicated when you go into other methods of delivery, say biolistic bombardment, because you can have fragmentation of the DNA vector that you're delivering into the plant. Hence, it can be more challenging to detect tDNA or I guess, your transgene using PCR. And in that case, you might have to resort to something like whole genome sequencing to really be sure that you don't have fragmented backbone, in your plant genome. But in my case, I'm just using agrobacterium mediated transformation for the most part. Amplification of, a small portion of your tDNA that is specific is sufficient to detect the transgene.
Okay, and then CJ, what's your process for detecting transgene free?
I think pretty much what Josh just mentioned. I will just add that you want to design multiple PCR primers, maybe on the right border side, on the left border side, and even in the middle. Because even agrobacteria, we also use agrobacteria transformation, and sometimes the tDNA could get truncated. So just multiple confirmation on both ends will be most assured. And then I think the gold standard is whole genome sequencing. You don't have to sequence deep. Illumina, Shori sequencing, shuttle sequencing to survey tDNA sequence present. It's actually an inexpensive way of confirming yes or no. You don't know the insertion site, but you can at least eliminate sequence traces.
Thanks. So I just want to take a minute and remind everyone about how I'm going to moderate the questions. So please type your questions in the Q&A box. So at the bottom of your screen, there should be a little box with a question mark. It'll say Q&A. I'm going to be moderating the questions through there, not through the chat. So please make sure that you're asking your questions in the right location. So with that, CJ, I'm going to direct this question to you as you have a bioinformatics heavy background. How many reads per sample do you usually get? And I think kind of the better question here is maybe not how many do you usually get, but what is the range that you're looking for to ensure high quality?
So we typically use Illumina short read sequencing. We do MiSeq. So at least here in our facility, we're talking about maybe $700, $800 for a library or MiSeq run, just so people can get an idea. It generates 1 million pair and 250 base pairs. So your amper count can be up to like 450, 470 base pair or so. So they kind of confine your search space, whether or not you have multiple copies, do you have SNPs and such. So that is our guiding principle. If you get 1 million reads, we usually pick 2,000, 3,000 alleles. And pay attention, I'm talking about alleles because you have to know, like if I'm targeting duplicated genes, 2 genes, 4 alleles, and then if you have 10 events, that's 40 for that genes. And then we pour across the lab, different project, even different species, none of those matters. You pour them together with barcoding. And then we just do a library when we have about 1,000 to 2,000 alleles. So if you do the math, they give you like 500 to 1,000 reads. And give or take, because our protocol, you don't have to be precise. We basically run a gel across, hundreds of samples and just eyeballing the band intensity, and they work perfectly fine for this purpose. So you got a lot of wiggling room. In practice, if you have 100 reads per allele, you are probably good. But then it get complicated if you're talking about duplicated genes and other issues. There are facilities, commercial facilities that accept amplicon sequencing samples. They charge less, but those libraries need to have barcodes already ready for sequencing. So that means you have different options of ordering your primers. We usually do two-step PCR. The first PCR is gene-specific primer with tail that is used as the binding site for the second PCR when you add barcodes. You can also order long primers that have gene-specific parts and then the barcode plus flow cell adapters, but primers will be more expensive that way. So there are multiple ways of approaching this. There are also long-read services like Plasmasaurus. You can submit PCR fragments. This could be up to 2 KB or 10 KB if you can get them to amplify. And especially for ploid, highly similar sequences, the short Illumina reads of 400 base pairs, you may not have SNPs that differentiate the alleles, then you can expand your space. You can send a PCR fragment, depends on the service level. You get, I can remember, 3,000, 5,000 reads. And then again, you do the math. If I have, 10 copies, 20 alleles, 5,000 reads, whether or not you get enough coverage for your sample. In those cases, you're talking about maybe $15, $30 per sample. So you can do some kind of funneling down, initial screening, and then certain cases you go for long read sequencing. It's probably more than what the initial questions are, but I think those are some of general guidelines. I think when CRISPR first started, many people rely on PCR. I saw several questions, but PCR really does not have the resolution to tell you exactly what the mutations are. When you have single base pair, two base pair, insertion deletion, PCR fragments are not gonna tell you the difference. You can design multiple guide per genes that's convenient for assay, but it actually cause a lot of complication in, actual application. So single guide per target size, the best method is to go for NGS sequencing.
Okay, great. So that did answer one of our questions. Is the DST PCR sufficient to confirm a gene knockout, or is sequencing still required? So definitely yes, you need to sequence. And you also touched on another question. This one was from Jeremy. Nanopore versus Illumina. And he's interested in the read number, the error rate, and the cost. So I think you mentioned it's about, you said it was like $30 per sample. I'm assuming that's for the Illumina. And then obviously this is going to differ based on, country and things.
Illumina, what we are doing is per library. So if you pack several thousand alleles or gene in the library, the per sample cost is really low. Although you are paying, $700, $800 for that assay, right?
Yeah.
For the long reads and PCR fragment, I think it's $15 to $30 a sample. You could design primers with barcodes, then you can actually pack multiple samples into one sequencing sample for service providers like Plasmidsaurus. I think many other Eurofins and others are also providing similar kind of service. I don't know if Josh has other things to add.
Yeah, I can comment on that. So I think our lab uses a similar approach. We do amplicon deep sequencing. So let's say you have 20 transgenic events, putatively they're edited, right? So you would first, with the first round of PCR, you would amplify that target site. And the PCR primers that you use have both a target-specific binding portion and also a bridge sequence at the 5' end. And the purpose of that bridge sequence is for amplification during a second round of PCR, whereas the second round of primers has specificity to that first round bridge sequence, right? So you can think of it as a nested PCR. And what we add on to that second round of primers are actually barcodes, forward and reverse barcodes. And so what we then do is say you have 20 transgenic events, right? And you've amplified each of them, and each of them, according to their primers, have unique barcodes contained in their amplicons. We then pull those together into one reaction, and we send it for Illumina Amplicon Easy sequencing. In particular, we use a company named GeneWiz, very famous company, and that's about $55 a sample. And we've also added on to that second round of primers an Illumina adapter sequence. So it basically saves a step for the sequencing company, and it also lowers the cost by about $30. So it goes, if you add the Illumina adapter sequence, it goes from about $80 down to, I think, $55 for that sequencing reaction. And then that basically, they provide us with paired end reads after that, right? So forward and reverse reads, you can think of it. And then with that, you merge those reads together, and you can use something like Flash. And then from there, we have all of these sequences that have been merged together, and they have barcodes, right, on their forward and reverse ends. So then we have to separate those sequencing reads according to their barcodes, because those barcodes belong to specific transgenic individual plants. And we separate them using a tool called CRISPR-Match. You can think of it as splitting the samples or demultiplexing them. And once you demultiplex those samples, we get anywhere from 5,000 to 8,000 reads per sample. And that obviously scales down the more samples that you add into that pooled sequencing reaction. So if you add like a full 96 well plate of samples and you pull them together, you get far fewer reads, maybe like, a few hundred reads per sample. And if you go as low as say like 20 sequencing samples and you pull that together, then you'll get more into the thousands of reads per sample. And from there, we go through the typical NGS analysis pipeline. There are different tools available: Argen-Cas analyzer, CRISPR-SO2, um, and then we can actually analyze the editing outcomes after that. So that's just an idea. So also the the question related to long read sequencing. So nanopore, you can send a sample like a long amplicon exceeding, say, 500 base pairs to Plasmidsaurus, and I believe they will sequence it for $15. But that's about 1,000 reads. If you request they have a different service, which is higher depth, and it gives you actually, I think, like 10,000 reads. And I think that's $30. So one thing about Nanopore is that the analysis tools available for Illumina and Sanger just aren't really out there. There have been a couple that have come out in like very recent months. So it wasn't until very recently that we have tools for analyzing nanopore sequencing outcomes. And I was actually working on a project where we had to look at amplicons of 6 kilobases in size. So we had to resort to nanopore sequencing. And at the time, there was nothing available to get the sort of quantification of edits and visualization that we often get with Sanger and Illumina tools. So I collaborated with a lab in Europe, and we produced a tool that allows us to analyze nanopore sequencing outcomes. And at the time, I think we were actually the first to do it, and it'll be published as part of a paper that we'll have coming out in the next year or so. But there was, I will say, I think just last month, the lab that produced CRISPR-SO2, the Pinello lab at MIT, has released a tool for analyzing nanopore sequencing data, and this is called CRISPR Lungo. As of now, I think it's just a preprint. But if you have nanopore results and you're unsure of what to do with it, you can look into that. Yeah.
Hey, thanks. And you kind of touched on you guys have been great speakers. You're like answering the questions as I'm going to ask them. So, you touched on CRISPR Espresso. CJ, I mentioned earlier you use your program, the AGE-seq. Both are using these pooled amplicons, right? And barcoding. So I have this question from Prathama. What are the standard techniques to confirm the edited plants? I have sequenced the edited lines, got some edits. How do I proceed to know how many copies have been edited in a polyploid crop with copies ranging from 10 to 12 for a single gene on a whole genome level? So, I just kind of want to start, you guys both have touched on these different techniques. I would say that there is not a standard technique. Everyone is using different methods, and we've already talked about some of those methods. So we mentioned RipFlip. There's the DST PCR. These are great starting points before you get into the sequencing, which we mentioned that's very important. But CJ, maybe we can start with you about what's the workflow for your lab?
Specifically for the questions or for, as I
So it looks
mentioned,
like Prathama is asking about editing in a polyploid crop. So her question was,
mhm.
what are the standard techniques to confirm the edited plants? I've sequenced the edited lines, got some edits. How do I proceed to know how many copies have been edited in a polyploid crop with copies ranging from 10 to 12 for a single gene?
Again, I guess the question is whether or not you have a reference genome. Congratulations, you got some edits, so you probably are in the neighborhood. I would suggest for polyploid, probably the best you can do is to design multiple set of primers. Again, you want for our empirical analysis, we want the primers to be conserved across your 10, 12 copies. So if I'm knocking out a gene family of 4, I want to look for regions that's conserved. You can include degenerate bases, so you can amplify multiple copies from the PCR. And then send them out for sequencing, especially for polyploid. I think the options of like Plasmasaurus that you can design 2 kb, 5 kb empiricon, especially for polyploid, your searching space should be a lot longer. And then $15 get you really probably 2,000, 3,000 reads that should allow you to differentiate between the alleles.
Mhm.
The common problem is your primer design might be biased. You might only amplify 6 or 8 of the 10 or 12. That is really, we are guessing, even for us having a high quality reference genome, it's not totally A approved. And there could also be bona fide somatic mutation in different lab strands and such. So sometimes you want to design multiple pair primers to confirm. As for our own practice, I kind of mentioned a little bit earlier, we just directly go to empiricon sequencing. We screen 10 events in our case, design consensus primer for the target we want to capture. Sometimes we cover on target and off target. If you have duplicates and only this gene you want to target, you want to actually make sure the off target is really are not edited. So the target can be specific, but empiricon primers for the assay, you can capture multiple copies. We sequence them, explore the libraries that Josh and I both mentioned. And then from there, we do a validation from selected subset of events. I think the case might be more complicated for cases that you don't have high editing efficiency, you get chimera and such. So those you probably require some tissue culture, subsequent screening to generate, or if you're working with annuals, you can go to next generation and have segregation to identify the best edited lines.
Yeah. I can maybe comment further on that. So polyploids are challenging to both edit and genotype, right? So you have a couple of options in terms of amplification of your target sequences. CJ mentioned, I think, using degenerate primers, which kind of act as universal primers for amplification of all of your copies slash subgenomes. So if you're looking at something like wheat, you have the three, the A, B, and D subgenomes. You can use primers that are universal to each of them, and then you can amplify all of those copies in a single PCR reaction. Now, one drawback to doing that is that you might have some PCR bias. It could be biased towards the A subgenome. Maybe it's a little bit, the amplicon's a little bit shorter, so you have amplicon bias as opposed to the D subgenome. So the way I approach this, because I don't want to have to deal with the downstream bioinformatics of trying to figure out the true editing efficiency of each of the subgenomes, I just use, and this works for me in wheat, I use allele specific primers or subgenome specific primers, right? So if you compare the A, B, and D subgenomes, you'll notice single nucleotide variations across them for the same gene, right? And if you just design it so that your primer has a mismatch at the 3 prime, the furthest 3 prime position, then you'll only amplify that specific copy that doesn't contain the mismatch. And for me, that works because I don't have to worry about PCR bias or downstream analysis. I can say, well, I'm only amplifying the A subgenome, right? Now, if you're working in something like strawberry or sugarcane, the genomes are even more complex, and so I wouldn't quite know. And that often the reference genome is not adequate, right? So it can be a lot more challenging. And really, what you have to do is, if you're interested in looking at subgenome specific editing, then you have to know how to separate those subgenomes and determines which ones are being edited or which ones aren't being edited, and you do that based on the presence of single nucleotide variations in those subgenomes, right? So you have universal amplification or subgenome specific amplification, and it's just going to depend on your context to figure out which one you want to use.
Thanks. So I'm gonna move on to our next question. This from UN. Do you pre-screen your T0 plants before sending to deep sequencing? If so, what's your screening method? And
Pre
I'll start with CJ for this one.
Um, I guess pre-screen in our case is antibiotics resistance from the tissue culture transformation process. Again, this is based on trees. I think we do have a little bit of work with Arabidopsis. If we are doing stable transformation, flower dip and all that, I think the first thing to do is to also do antibiotic screening to make sure Cas9 is there. I know this is not the preferred way when you talk about gene editing, but we're talking about standard editing experiment. You want to make sure your tDNA is there, and then from there you select random events for your sequencing analysis.
Yes, and it's the same for us. We rely on, antibiotic selection, and then we pretty much go straight to sequencing.
Mhm.
So if you want to pre-screen and your guide RNA happens to, say, abolish a restriction enzyme site, then you could do RIFLIP as a pre-screening method. Generally, I think it's more cost effective and time saving to just move forward, yeah, to the
Mhm.
sequencing.
Okay, thanks. Next question from Han Wei. Hello, I get sequence results from Plasmidsaurus using nanopore tech. It shows some mutation has 50% reads or over, but there is a very low wild type reads, lower than 10%. Does this mean this mutation is a homolog mutation? CJ, I'll start with you for this.
It depends on your target organism. Is it diploid and single gene or multi-genes? But I can say that if you don't see wild type reads, assuming you have your wild type samples in, separate analysis, then you probably have editing. Whether or not it's by allelic or homozygous, it really depends on the mutation, right? If you're only seeing one type of mutation or two types, and then also the term in the CRISPR field, in genetics, biallelic means mutation in two allele. They don't need to be the same. It could be plus one, minus one. It's mutated in their gene in both allele. Whether or not you have homozygous biallelic or heterozygous biallelic in terms of as far as function is concerned, if there are loss of function, friendship mutation, you have mutation. But I guess in your case, it's hard to really answer that. You should be able to see two different kind of mutation unless for sure indels, many of us have seen you see plus one all over the place. And then in that case, you might have a homozygous edit.
Okay, thanks. Josh, did you have anything to add to that?
Yeah, basically the same thing. If you know that there are additional copies present of the gene, then you need a way to rule out that you're not looking at like the your wild type reads aren't just background copies of that extra homolog that exists. But one thing we often see is I think they mentioned over 50% of their reads are edited, something to that. And then I think they said, could you maybe repeat that part, Amy?
Sure.
Yeah.
Um, Sorry, let me go back to the question.
Yeah, well, just for
Yeah,
reference.
sorry.
That's okay.
I get sequence results from Plasmidsaurus using nanopore tech. It shows some mutation, 50% reads or over, but there is very low wild type reads, lower than 10%.
Oh, yeah. So it's not uncommon to get either contamination wild type reads. I mean, PCR is really, really sensitive. It's and that things can happen during the sequencing as well. There could be contamination there in the pooling the samples if you happen to be doing that. So they could be background contamination reads. And the other possibility is that not all of the cells in your population from which you extracted DNA are actually edited, right? So you might have some mosaicism or chimerism, and that's contributing to the background amplification. And the only way you can rule that out is to go into the next generation, right? So you'll either get, wild type or heterozygous or biallelic in the, the next generation. You can't really rule that out otherwise. But generally, it's a good sign if you're seeing excess of 50% editing efficiency with lower than 10%, wild type reads, I would say.
And that actually brings me to so someone had a question about carrying the next generation. So this is from Easter. I obtained about 98% efficiency in T0 wheat lines. These were prime edited using CAPS and Sanger, which was analyzed via titer. But this dropped to about 50% in the T1 generation. This is with the T0 selfed. Could this be due to T0 chimerism? Would NGS have captured low frequency edits better for predicting heritability?
Yeah, I can
And then
comment
Vina has
on
a good
that.
point that many of the reasons for this observation you described, so the chimerism,
Mhm.
but I'll let you comment on that. So, Josh, maybe we could start with you on.
Yeah, I mean, if they're not following the expected segregation ratios, then that would suggest that your edits failed to be inherited. And a great model for looking at this phenomenon is actually Arabidopsis. So in Arabidopsis, people think of it as easy to, it is easy to transform, of course, but to edit, it's actually not that easy to get high efficient genome editing in Arabidopsis. So people use, constitutive pull two promoters and see a high frequency of somatic edits in say the leaf tissue, and those edits will not be inherited in the next generation. So what people do to ameliorate that is to use egg cell specific promoters, and then that results in activity of your CRISPR, editing machinery specifically in the egg cell or in the zygote, and that, there's fewer copies present to edits there, and then it develops into a fully edited offspring. So basically, what they're describing in my mind is just somatic tissue editing in the T0 generation, and that those edits aren't being contributed to the germline, probably as a result of genetic mosaicism, which Vina mentioned. Yeah.
Thanks. Yeah, that's a really good point. So the somatic editing does not equal the germline edits.
Inherited. Yeah.
CJ, do you have anything to add?
Yeah, I will echo the suggestions already that CAPS and other PCR-based assays do not have the resolution. So if you can go for sequencing-based analysis, some of those minor edits will probably be better.
Great. So we'll move on to our next question. This is from UN again. So it's just a clarifying question. It sounds like both of your labs take a strategy of screening transgenic plants carrying Cas9 by antibiotic selection, then conduct deep sequencing on the transgenic T0 plants by pooling 10 to 20 events. Generally, you target at thousands of reads, Illumina short reads, or hundreds of reads from nanopore per event to check the editing profile. At last, you will confirm the edits in the individual events by Sanger sequencing. Do I understand correctly? Yeah, so I think everything was good. I saw nods right up until that last part. So, CJ, can you clarify?
Yeah, no, we don't. I think the pulling part, pulling many, many hundreds of events in our library, those are all barcoded. So when you get the data, you have the barcodes, you know which event has what added that you see. And then the only thing that we do is we confirm, we select the events that we want to propagate and for characterization, and only those that we confirm second time by empirical sequencing. We do not do Sanger simply because in the complex genome, when you have more than two alleles, Sangers really do not have the resolution.
Yeah, it's same for us. So you have the pipeline nailed down right up to the very last step where you mentioned Sanger sequencing. So doing an additional round of Sanger would be sort of redundant because we've already seen to the fullest extent what the edits are from our pooled NGS, right? We know what each individual event has for the genotype, right? So there's no need to do Sanger sequencing there.
Great. So thanks for clarifying that for
Mhm.
the audience. So I have two questions that are similar. So I have one from Rakesh and then one from Vina. So Vina, this is more of a comment, but can you please comment on the significance of checking and reconfirming edits in future generations to ensure the plants you are working with indeed have the edit of your interest? CJ, I'll start with you.
Yes, I think part of this depends on the regeneration workflow. In our case, we do, like I say, we do a second round of confirmation. For the most part, we get reproducible data. Occasionally, we see variation, and I ascribe this to be usually, like I say, we go through a catalyst and then organogenesis process. Typically, if you isolate the events early, every shoot from the caddis clump should be, identical, but sometimes they are not because they could be from two adjacent transformed or edited cells. So they could just be that two sampling came from two different events. Our routine is we propagate only from a single individual in tissue culture. We don't take multiple shoots from the climb just because of potential thing. And it ended up spending way more time to figure out all the disagreement of data. So confirmation is important. And then in academic labs, when we have so many undergraduate student helper, and tissue culture is a very labor-intensive process, a common error is human error, labeling and such. So yes, before we launch into very expensive greenhouse or field data, it won't hurt to reconfirm again.
Yeah, and I think she mentioned future generations. I just to nail down the point on this. You can observe editing in the T0 generation, and that isn't necessarily inherited into the T1 generation. So that's the importance of screening your T1 events to actually confirm inheritance. You can't just assume that your edits will be inherited if they're present in the T0 generation.
And in addition, I guess unless you segregate out cassette, you
Yep.
will get new edit, right? So I
Oh
think,
yeah. Yeah.
once you get stable, the edit you want, I think the next step for annual
Mhm.
that you want to go to the case or TDNF-free generation, so your edit will be fixed and will not be changed or, introducing
Yep.
additional complication.
Yeah, if your target site co-segregates with the presence of Cas9, then you can have ongoing editing in that next generation. And you can even see, as a result of that, even some mosaicism in the T1 generation, which could be very perplexing. So there is ongoing Cas9 activity dependent on its presence after segregation.
Great. Thank you both for that. So, next question is from Ling. I would like to know which source gives the best sequencing results. I've used the University of Georgia sequencing facility, Eurofins, and Plasmasaurus for Illumina NGS and Nanopore, but the sequencing result quality seems to vary many times. Thanks. So, yeah, I think I'll just start with this. This is you're always going to get variation in the sequencing quality. There's just so many factors, but I'll turn it over to CJ to talk about some things that be affecting your quality. But you're not
Sorry,
alone in
my
this.
local colleagues here, I use the same facilities. I don't have problems per se, except for occasional flow cell failure, then the whole
Mhm.
data, whole library was lousy. But because we usually pull hundreds and thousands of samples together, I often see one gene failed. The others were fine. So there's a lot of sample to sample issue, just typical PCR issue. I don't think that it's one size fits all. Josh mentioned the gene width offer of the amplicon sequencing. That is great, but we sometimes get kind of a little bit mixed data because those facility pull samples from all users. So sometimes contamination does happen. So it, yeah,
Yeah.
we can talk offline. I think if you are getting inconsistent data, I suspect it's more in the wet lab parts rather than the sequencing process per se.
Yeah, I was just going to comment. So what are some things that are in your control that can help, to produce higher quality sequencing results? A number of things. I mean, if you're doing pooled amplicon sequencing, you want to have a highly specific and very abundant amplicon, right? So not all of your target sites are going to amplify at the same level. So obviously you would expect to see some level of variation in the quantity and the quality of your reads, if you don't have consistency there. So that's one thing. And, if you're doing whole genome sequencing, the quality of your DNA extraction matters a lot. And, if especially if you're doing something like long read sequencing, you need to extract high quality, high molecular weight DNA. And using CTAB often, isn't sufficient. You need to use some kits which are generated specifically for doing these sorts of experiments. So aside from just the maybe mistakes on the sequencing aspect, doing everything leading up to that part to ensure high quality DNA extracts can help you a lot. Yeah.
Great. So this is a question. Is there a well-accepted definition of the term editing efficiency? So I would definitely say no, but I'll let you guys turn it over to what is, yeah, so the term editing efficiency is used every manuscript, but I think what is considered efficient is debatable. So Josh, I'll start with you for this one.
Yeah, I agree. I don't think there's an exact definition of editing efficiency because when you're examining editing efficiency, uh, you always have bias, right? So, for example, PCR bias. If you amplify a target site, um, your percentage of modified reads can be biased based on how that target site amplified. So in particular, here's how we define editing efficiency. After we amplify a target site and we get the reads from that and we edit it, or we examine the edits, we would define it as the proportion of modified reads. So the proportion of reads that have any sort of, an insertion, a deletion, sometimes substitutions, where you would expect it to. So in the case of Cas9, typically, so Cas9 cleaves at say position 17, between 17 and 18 in the 20 nucleotide guide RNA. So you would expect to see indels, insertions and deletions, within the surrounding few base pairs around that, right? So you wouldn't consider like a substitution mutation at the 5' end of your guide RNA to be a Cas9 induced mutation, right? That was probably just sequencing error or something unrelated to editing. Um, but the way we would define it is, yeah, a proportion of modified reads. So if you have 10 reads, of course we deal with thousands of reads, but just to make my math simple, if you have 10 reads and four of them contain an indel of some sort, then your editing efficiency would be 40%, right? So that's just an example.
Thanks. Yeah, I think the efficiency, it's like I said, there's no set definition or standard for that. So this question is from Max. In the system I work with, the mutations in the T0 generation rarely are inherited to the T1 generation. I've stopped analyzing the CRISPR target in the T0 generation entirely. Why do you spend so much effort in analyzing the T0 generation when you have to repeat the process in the T1? CJ, do you have any thoughts on the inheritance here?
Um, Again, I agree. I think, you select based on antibiotic resistance and go to T1. But there are also approaches, especially intronized the Cas9, for example, that greatly increase the T0 editing efficiency. So I think it's context dependent, depends on your system, depends on, just the overall efficiency in your lab when you are working on those. If you have such a low efficiency in T0, yes, I think it makes sense to, jumping into T0, T1 and then spend efforts there. But you do need to repeat.
Josh, any additional thoughts on
Yeah.
this?
So that's a really good question. So basically, it's why even bother looking at T0 genotypes if you're really only interested in, I guess, the inherited or the T1 and events. So you might get a lot of T1 seeds, for example, from a T0 event that isn't edited or has a very low editing efficiency. So that would be sort of a waste of time in looking at the T1 event if your editing efficiency is so incredibly low in the T0 generation. So it's more efficient to look at your individual T0 events, maybe select the ones that have 50% or 40% or above editing efficiency, and then move those forward into the T1 generation and examine those events, right? So it's more of a time saving feature. And as CJ mentioned, adding introns, so like to Cas9, for example, there's a very famous enzyme called it's a maize codon optimized ZCas9I. It contains 13 Arabidopsis introns, and that seems to enhance the editing efficiency in dicots, and I think maybe even recently it was demonstrated in monocots. Um, yeah, so that's one method that you could use that nuclease to bolster your T0 editing efficiency, making it more likely to obtain edits in the T1 generation.
Yep.
Great. So this is a question from Essie. I have Cs for my T0 plants, which do not show any edits despite PCR confirmation of Cas9. What would be the next steps in such a case?
It's tough to say because the Cas9 PCR confirmation, it could have been truncated and edited unless you get a whole case. If you want to continue, you should try to get different independent event. You have seeds from T0 plants, so assuming those are different individual events. But yeah, there could be many different things.
Yeah, and so what are some parameters that could result in low editing or zero editing efficiency? Poor guide design. You need to make sure that you're using the right strand and there's the presence of an NGG PAM. That's very basic, right? But some guides just, they don't work for genome editing, and that probably has something to do with the chromatin context of your target site. So you could have a perfectly designed guide RNA and you just cannot edit that gene. It probably relates to, the lack of Cas9 accessing the target site. Some target sites are highly, condensed chromatin during different developmental stages, making it difficult or challenging to obtain edits of those sites. So it's not always your fault per se, but always check to make sure your guide parameters are good. You want your guide RNA to be specific. You want it to have, say, 40 to ideally like 60% GC content. You can go a little higher than that too. The presence of a PAM. So those are some things to consider. And don't always just assume it's a mistake that you've made. It could just be a challenging to edit target site.
Great. Yeah, thank you for summarizing that. Like you said, there's many things that could be going wrong there. Those are just some of the things that to look out for. So we are almost at the end of the hour. This is going to be our last question. This is from Lucas. Could you both summarize the tools slash services you use for barcode design, amplicon NGS sequencing, and sequencing slash edit analysis?
I can start. We are doing two-step amplicon library preparation, so we're still using the very old-fashioned Illumina barcode primers. Those are publicly available, many different sets. So that is standard. Empirical NGS sequencing, again, mostly standard Illumina amplification, two-step amplification process. Sequencing edit analysis, as Amy mentioned, we have our kind of open source HSeq workflow. It's self-executable as long as your computer has a Perl script and install the blade. Anybody can run it. I'm not a bioinformatician. I can run it. Undergraduate student in the lab can run it. So there are issues because it's 10 years old now. And then Windows, every new operating system sometimes introduce a little bit of the glitch. But there are many tools now out there that you need to have your reference sequence as the guide, and reads are mapping to the reference sequence to sort out the editing pattern. So often the complication is if you don't have the right reference, it's garbage in, garbage out. You're gonna get, complicated data.
Yeah, and for our primers, I'll just refer you to an article or a tool, rather, you can think of it called HiTOM, which stands for High Throughput Tracking of Mutations. And that article will provide you with the exact primers that we use in our second round of amplification. They're bar-coded primers. So you have a 96 well plate, and each well in the 96 well plate has a different combination of forward and reverse primers, right? So I would just check out that article. And then going beyond that, we pull those samples together. We send them to GeneWiz typically for Illumina Amplicon Easy sequencing. And then there's a few different options for analysis downstream of that. But just in brief, I merge the reads using Flash. I demultiplex or split the reads using a tool called CRISPR-match in the command line. It has a graphical user interface though. So once you install it, it's simple to use. And then I tend to go to Crispresso 2, which has a web page as well. I do that in the command line, and that's how I quantify my edits. And then I usually will visualize my edits and make graphs and things using R, but you can also use even Excel, right? So that's kind of the whole standard pipeline there.
Great. So we are just over the hour, so I'm going to close out the session here. Just first, I want to thank our speakers so much for their time, their expertise. You gave such great advice to the attendees here. I think this is really practical knowledge that people can go and apply immediately into the lab to support their research. So just thank you. And I see some hearts coming up on the screen. Just I know your time and your expertise is appreciated by the community. I know we didn't get to answer everyone's questions. I do want to remind you that we have a discussion forum. So if you are a PlantGene member and you log into the website, you will be directed. There's a page that's going to come up. You can go to our collaboration hub. You can go to our discussion forum. I really encourage you to ask your questions there. I haven't seen a question gone unanswered. People who have reached out through the discussion forum have always gotten a response. So please do ask those questions. I saw some people asking for the recording. This has been recorded, and I will be sending those out. Again, PlantGene members get emails from me with all of the recordings and event updates. So if you are not a PlantGene member, please head to our website and register. Our website did experience an outage this morning. I've been told that everything should be up and working. That wasn't on our end. That's just with WordPress in general. But if you do have any issues, just maybe give it a little bit of time. If you're still experiencing some issues, please send me an email and I can help you troubleshoot. So again, thank you guys so much, and I will see you at our next event. All right.
Bye.
Bye. PlantGene would like to thank the National Science Foundation for their support. To learn more, please visit their website at nsf.gov.